Good night. Our group is composed by David Eduardo Barros Flores and Daniela María Chaparro Carreño, and we will expose to you about the diagnosis of pigmentary anomalies in the bird population of the Rallidae family present in the Juan Amarillo, La Conejera and Jaboque wetlands of the city of Bogotá D.C.
Overall objectives: diagnosis of the presence of pigmentary anomalies in the birds population of the Rallidae family present in the Juan Amarillo, La Conejera and Jaboque wetland of the city of Bogotá D.C. Specific objectives: estimate 3 to 4 species of the Rallidae family predominant in the 3 wetlands selected, through monitoring and data collection in ecological corridors. 2. Identify the specific gen that define the pigmentation and anomalies of the plumage, beak and legs according to their phenotype and genotype characteristics. 3. Record occurrence of individual white alteration in the color of the plumage, beak or legs, and their relationship with wetland considered contaminated or conserved. 4. Highlight the ecosystem function that the Rallidae family plays in the wetland ecosystem conditioned to the urban context. There are multiple alterations associated with melanin pigmented plumage. Among the best known we can find albinism. In albinos, a mutation possibly affecting the enzyme tyrosinase completely inhibits the ability to synthesize any type of melanin, and on the contrary, in melanins, the synthesis of melanin is overstimulated and a production and deposited of melanin occurs where it normally should not occur. The metabolic route of melanogenesis. Melanin is synthesized by the body itself in specialized cells called melanocytes. They become the neural crest and migrate to the epidermis of the embryo, near the germinal center of the future feathers. When the time comes, the melanocytes activated their cellular machinery and behind to synthesize melanin, which is stored in the organelles called melanosomes. Melanosomes are spherical when they contain eumelanin and oval when they contain pheomelanin. The melanocytes are collected next to keratocytes, the cells that produce the keratin that forms the feathers, to which they transfer the melanosomes, which are deposited in the keratin fibers of the pen during their growth. Pigmenting it desynthesized is given by complex metabolic route. Unlike what happened in mammals, the genetic basis of the melanin synthesis process in birds is still in an initial phase of study. In the late 90s, a gene that encodes a protein called melanocortin 1 receptor located in the cell membrane that seem to play a central role in regulating melanin synthesis and the time of the tilting say synthesis towards EU or pheomelanin. The melanocytos precursors flow from horizontal rings at the proximal base of the feathers follicles and migrate to the barber rigis and produce pigment during feather growth. The genes that interest use in the regulation of synthesis of melanin production are melanocortin receptor 1 emits the necessary signal to diverse production towards pheomelanin in the species that have been studied until the date the melanocortin receptor 1 alleles that cause increase in the synthesis of eumelanin are inherent as dominant, while that those that cause an increase in pheomelanin as are usually recessive. Tyrosinase gene. The tyrosinase is the limiting enzyme for melanin biosynthesis which occurs in melanosomes within melanocytes. The tyrosine and tyrosine-related proteins type 1 and 2 are involved in the production of eumelanin. Only the presence of cysteine and some tyrosinase activity may be necessary for the production of pheomelanin. When tyrosinase is expressed at low levels, pheomelanin is produced by adding cysteine to dopaquinone. The next gene is SLC45A2 gene. This protein is associated with the variation of pigmentation in several vertebrates. It was previously known as membrane-associated transport protein, but the function of SLC45A2 is not fully understood. Mutations in SLC45A2 have been known to disrupt tyrosine processing and traffic at the post-Golgi level.
Phenotypic identification of Rallidae species. In the realized sighting, we found 3 species from Rallidae family. There were Fulica americana, known as Focha común, Porphyriops melanops, known as Tingua moteada, and Gallinula galeata, known as Tingua pico rojo. As results, in the Jaboque wetlands sighting, we found a specific richness of Rallidae of 3 species. The most numerous species was Tingua pico rojo, with 33 individuals. In the map of the sampling area, it is possible to observe that the distribution of species was sited in different microecosystems, such as Tingua moteada, that inhabits stagnant water bodies, or Focha común, that inhabits open water bodies. Also, we cited interspecific relationships, such as Tingua moteada and Tingua pico rojo share the same habitat, or as Tingua pico rojo share the area with herons. At the bottom right, there is a photo of a young Tingua pico rojo. About environmental conditions of Jaboque wetland, we can highlight that the ecosystem has recovery interventions by removing invasive plants and deepening the lake to about 3 meters deep with the help of heavy machinery. It presents a considerable amount of plant species and fauna of both birds and rodent mammals. We can find that the presence of rubbish in the bodies of water, such as tires, debris, and plastics. There are still sewage pipes that they discharge that polluting materials into the wetland. And it was observed that the proximity to the river Bogotá has an impact on the environmental conditions of this area. As results, in the Juan Amarillo wetland siting, we found a specific richness of Rallidae family of 2 species. The most numerous species was Focha común, with 37 individuals observed. In the map of the sampling area, it is possible to observe that the distribution of species was sited in different microecosystems, as Tingua pico rojo in the less intervened area, or Focha común that inhabits open water bodies. In this case, were not observed any Tingua moteada. In Juan Amarillo wetland, it was observed a family of fochas or coots close to its nest. The pattern of color was closely observed in coots, finding a bird with white beak as it is observed in the photo. About the environmental conditions of Juan Amarillo wetland, we observed that it presents a considerable amount of plant species and fauna of both birds and rodent mammals. A lot of water hyacinth present in the wetland can affect the life into the ecosystem as eutrophication. We observe the presence of rubbish in the bodies of water, such as tires, debris, and plastics. Also, the proximity to the river Arzobispo has an impact on the environmental conditions of the area. And finally, it was observed livestock near the river. To compare the results between Juan Amarillo and Jaboque wetlands, we use some diversity index as Mergalef and Simpson. Mergalef index serves to estimate the biodiversity of a community where at values greater than 5 it is considered a high biodiversity in the area, while value is below 2 are considered to be an area with low biodiversity, such as Juan Amarillo wetland result in DMG index refers a zone with low biodiversity in Rallidae birds under 2, with a result of 0.271. Also, Simpson index represents the possibility that one of 2 individuals into a same region and selected by hazard were from the same species. We observed that Simpson index in Jaboque wetland refers a media to high biodiversity in Rallidae birds near to 0.0 as result of 0.341. Comparing the results obtained by sighting into wetlands with the sightings from a lot of people filed in Naturalista webpage, we can observe that the proportion in sightings is similar. In the last sighting into Juan Amarillo wetland, we observed some Fólicas americanas that present despigmentation in their beaks, as is shown in photo. According to Rodriguez-Ruiz et al., leucism is a genetic, recessive, and heritable mutation which is caused by an error in melanin deposition. So we can observe in photo an individual or Fólica americana sighted in 2016 into Juan Amarillo wetland that present this phenotype that can be distinguished from albinism in the way that the bird with leucism presents some areas without coloration but conserve commonly colors in eyes, beaks, or legs. Aging presents symptoms like leucism, but it is caused by a disorder in a bird life period or induced by environmental conditions as contamination or food availability. So it is possible that the aberration found in the pigments of pig will be related with an aging symptoms, considering that, as Rodriguez, Casanova, and Surya researched, the variations in plumage color can result by genetic facts or environmental ones, principally in disturbed areas. These aberrations are expressed as negative selection of wildlife, which leads to a disadvantage because they are more visible to potential predators or creates difficulty in getting a mate.